Physiological classification of proteins by function. So what we're going to see is that the proteins located within the cells, within the tissues, within the organs, within the body are going to have a very dramatic role in controlling what is going on within the body. And so the proteins, again, based on their composition and their structure, are going to have many distinct functions within the body. Now, one of the first examples of protein function is going to be the enzymes, which have a role in controlling metabolism. Basically, what we see with an enzyme is we're going to have a relatively large protein, and through its shape interactions, that protein is going to bind to a substrate or substrates, basically bind to the reactants in this chemical reaction. The fact that we're binding to the reactants is going to bring those molecules close together, and it's going to promote the chemical reaction to occur. In general, it's going to lower the activation energy to make this reaction more likely to occur. In this case, we're taking a molecule and breaking it apart, and so we're bringing the molecule in to the binding site of the enzyme. So again, shape matters, charge matters, so we have that interaction between the enzyme and the substrate, and in this case, we're producing the products, which are going to be, in this case, broken down components from this individual substrate, which are then being released, and that enzyme can then circle back and control another chemical reaction of that same type of substrate. The next category of proteins are going to be membrane transporters, and these could be in the form of either channels or tunnels. And so what we're going to see with channel proteins is they are going to be transmembrane proteins that are going to span across the cell membrane, and basically to the inside of this channel protein is going to be a hydrophilic pore, a region where aqueous molecules, hydrophilic molecules, charged molecules are going to be able to pass through the membrane because they're basically passing through a tunnel, and so they don't have to interact with the hydrophobic characteristics of the fatty acid tails. They're able to move through the membrane through these ion channels or other types of channels. The other type of membrane transporters are going to be carrier proteins. In this case, as opposed to simply forming a tunnel or a tube through the membrane, we're going to be involved with physically moving molecules across the membrane. And so in general, what we're going to see with these membrane carriers, proteins, is they're going to have two configurations. One configuration that is kind of open to the inside or cytoplasmic side of the membrane, another configuration which is open to the outside or the extracellular side of the membrane. And then by switching between the outside opening and the inside opening form of this protein, they're able to move the molecules across the membrane, again, without those molecules having to interact with the hydrophobic characteristics of the cell membrane. The next category of proteins are going to be signaling molecules and receptors. We're kind of looping these things in together. So examples of signaling molecules would be things like hormones or neurotransmitters, and then we're going to have receptors that are capable of binding to these signal molecules and doing something with it. And so an example of this, if we're taking a look at a neurotransmitter, it's forming, the neurotransmitter would be the ligand here. The ligand is going to bind to an ion channel, and as a result of binding to that ion channel, we're going to cause an opening of that ion channel and a flow of ions down their concentration gradient. Another form of a receptor in response to a signal molecule are going to be these G protein coupled receptors. In this case, again, the yellow ligand is going to be capable of binding to the receptor, but what happens here is we're going to convert that external signal, the ligand here, in many cases a hormone, into an internal signal. And so this is basically going to activate a G protein on the cytoplasmic side of the membrane, and that G protein then is going to go through a series of signaling reactions that are going to result in some type of activity change within the cell. And we'll cover this in a different video. The next category of proteins are going to be binding proteins. Now, these binding proteins are going to be involved with changing the solubility of molecules so that these molecules can be transported through the body. Now, a good example of a binding protein would be going back to our hemoglobin molecule over here. In general, oxygen is not going to be very soluble within plasma, and so in the absence of hemoglobin, there is going to be very low levels of oxygen being transported throughout the body. By having this hemoglobin molecule present, we can bind to oxygen, actually 4 oxygen molecules here, and very efficiently transport oxygen throughout the body. Another example of a binding protein is taking a look at the hormone testosterone. Testosterone is a steroid hormone built from a cholesterol molecule, and so it's going to have hydrophobic characteristics. It's not going to be able to be dissolved well within the aqueous environment, the watery environment within the body and within the bloodstream. And so what we can see is that we've got transport molecules like albumin here, another example over here. And these albumin molecules basically kind of surround our testosterone, our steroid hormone here, and take what would normally be a hydrophobic molecule, kind of hide the hydrophobic characteristics by having an overall hydrophilic shell around the outside. And so this albumin then can transport testosterone throughout the body and then release that testosterone to the locations where it needs to be binding to a receptor and be picked up by a cell. Immunoglobulins are going to be the next category of proteins. These are going to be important for the immune response within the body. And so what we're going to see within an immunoglobulin or an antibody is 4 polypeptide chains coming together. We're going to have a constant region down here, which is going to help dictate what happens with the information by the the antibody binding to an antigen. And then we've got a highly variable antigen binding region up here. This is what's going to be variable. This is going to be able to bind to many different shapes because we're going to have many different very specific antibodies. And again, we'll cover this within the immune system. Regulatory proteins are the next category of proteins. These are going to be proteins that are going to be able to control the metabolism and other aspects of cell activity. And so examples of a regulatory protein would be things like transcription factors. We are going to have enhancers and transcription factors and mediators and promoters that are all going to go in, bind to the DNA, open up the DNA, and then allow for the kind of attachment of a polymerase. In this case, we're taking a look at an RNA polymerase. And so we're going in, opening up the DNA molecule to be able to make a messenger RNA copy. All of these regulatory proteins are going to be important for controlling what region of our DNA is going to be copied into a messenger RNA molecule. These other regulatory proteins can control other aspects of cellular metabolism and chemical reactions that are occurring within the body. Structural proteins are another example of proteins. They're going to be there to support the three-dimensional shape inside and outside of the cells. So they're going to be contributing to the shape of a body. And so if we take a look at this, we've got a cell membrane here. The lighter green to the bottom is cytosolic. This is going to be the intracellular inside of the cell. And outside the cell, in this case, we've got the extracellular matrix. And so we have these structural proteins in the form of collagen. We've got laminin over here, other molecules that are coming together that are going to contribute to the structural characteristics of connective tissues. Inside of the cell, again, we have structural proteins in the form of the cytoskeleton. And so we've got actin rods, we've got microtubules, the largest of these intermediate filaments. And then finally, we've got these intermediate filaments here in the middle, kind of intermediate in shape, distinct and unique based on different types of cells. And then again, they're going to provide kind of structure and support for the cells. And then the final example of protein function are going to be contractile proteins. And so generally when we think about contractile proteins, we're thinking about movement. And so we could be talking about muscular movement or movement of things within the body. And so we're seeing interactions between actin and myosin and all of the other regulatory proteins that are involved with this that allow for the actin and myosin to interact, shorten this contractile unit, and result in some shortening of the cell or movement within the cell or movement within the body.